So it is my great pleasure to introduce our first invited speaker, Dr. Melissa Cline. She's joining us today from the University of California, Santa Cruz, where she's faculty member in the Department of Molecular and Cellular and Developmental Biology. Her background is in computer science, and her research interests have been in studying alternative splicing, also significance of germline variants that are associated with cancer predisposition. She has done some very important work related to interpretation of BRCA1 and BRCA2 variants through her leadership roles in BRCA exchange project, which is one of the driver projects for GA4GH, Global Alliance for Genomics and Health. She has been very actively involved in some other initiatives through GA4GH related to data exchange, standardization, and she has worked on developing better approaches for variant interpretation, both for germline and cancer variants. So she will be telling us more about those initiatives in her talk entitled Federated Analysis for Germline Variant Interpretation. So thank you very much for joining us today, and we are looking forward to your talk.
Yeah. Well, thank you very much, Gordana, and I want to thank the organizers of the meeting for this invitation. So, with this talk, we're moving from treatment to heritable cancer susceptibility, and to first touch on some motivation. As many of you will know, a large fraction of tumors are driven in part by germline variants that put the individual at greater risk of cancer from birth. In some work here at Washington University by Li Ding and colleagues, looking at TCGA cohorts, overall, 8% of the individuals in TCGA carried some pathogenic or likely pathogenic variant that would could in part explain their cancer risk. And while the frequency varied by cohort, which is also shown in the literature, overall, that's about 1 out of 10 individuals whose cancer came in part from hereditary factors, and those factors can be significant. In BRCA1 and 2, in which I do a lot of work, we see that while the average woman has about a 1 in 8 chance of cancer in her lifetime, a woman who carries a pathogenic BRCA1 or 2 variant has a cancer risk that's closer to 2 out of 3. Now, these cancers this cancer risk can be detected in advance, and there are steps that women can take to practically eliminate that cancer risk. The choices aren't easy, bilateral mastectomies, but the patient has the choice. That is, the patient has the choice if the genetic risk is recognized, and that brings us to the challenge. Here, when we look at the variants, the cancer-related variants that are in ClinVar, we see that the largest group of them are variants of unknown significance. And these are rare germline variants for which there's just not enough evidence to make any definitive claim on their impact. Now, when we look at more detail in BRCA1 and 2, we see a similar proportion of variants of uncertain significance, VUS, around 40 to 45%, but we also see that's dwarfed by this huge number of variants in NOMAD that aren't even in ClinVar yet. And these are variants from recent sequencing studies, variants that were observed in actual human beings in recent times. So when you hear about the variant interpretation bottleneck, that's what it looks like. And saying a little bit about its impact, when a patient gets a report with a VUS, well, that can, of course, lead to uncertainty, worry. In some cases, it leads to egregious clinical mismanagement. There are horrible stories, all too frequent, about women who undergo bilateral mastectomies because of VUS, which may still turn out to be benign. But the major impact is that the patient who receives a VUS test report is a patient who does not have the opportunity to clinically manage his or her cancer risk. So with all of that said, how are VUS interpreted? Well, interpretation involves weighing together many different forms of evidence, but really chief among those are clinical observations of patients who carry the variant and their families. And it turns out that these observations are necessary for really most variant interpretation. Even if you have a really, really strong functional assay, variant curators will want to see the impact of the variant in a person to make sure that the cell in its wisdom didn't somehow correct for the impact of the variant, that the variant actually leaves a phenotypic effect. Now, this brings us to, well, the first problem that most cancer susceptibility variants are highly rare, and most of that data is siloed, that is restricted to one institution. And so putting those two things together, most labs won't have enough data for many years to interpret the rare variants that they see. And that means many patients who lack the opportunity for clinical management of their cancer risk. This would, well, it would seem that data sharing would be an obvious solution, and contrary to what one might expect, even commercial testing labs are most often highly collaborative and very open to data sharing because well, improved variant interpretation benefits the whole industry, the whole field. So what about data sharing? Why aren't we sharing more data? And the big catch is that the data that we need to look at is sensitive. It usually cannot be shared directly by a, well, complex landscape of regulatory, ethical restrictions. But it turns out that the data that's most important for variant interpretation isn't the individual records themselves, but statistical variant level summaries of those records, such as case control ratios or allele frequencies. So so then the problem becomes how to get those analysis results and how to move that analysis process to the lab easily. And this brings us to federated analysis, which we describe as bringing the code to the data rather than bringing the data to the code. In a traditional data sharing model, which, by the way, rarely works, a lab might share their data with analysis collaborators who will run their analysis software on it to generate variant level summaries, which ultimately make their way into ClinVar. In the federated model, the analysis software is instead shared with the labs to run on their own data, and then that produces variant level summaries which are much less sensitive and can be shared much more easily. So then that summary information is shared with variant curators who curate the variant and deposit the curation in ClinVar. We've had a successful proof of concept on this in collaboration with Raca, which holds a large cancer cohort from individuals from BioBank Japan. We sent them some analysis software. Their cohort, of course, is something that they would not be able to share with us directly, but they were able to run our analysis software on their patient data and give us variant level summary results, which we use to interpret a number of variants. Now, I'll show you a couple other examples of federated analysis. One is the Seneca project, which is doing beautiful work, combining data from Europe, Canada, and Asia in a sophisticated architecture that's allowing them to generate cross-cohort GWAS and allele frequencies. And then there's the Treehouse project, the beautiful project in which is about analyzing pediatric cancer and somewhat similar to the sort of analysis that Lucas was describing earlier. But it starts with RNAseq from a child's tumor. That RNAseq data is itself, of course, identifiable, not something that could be shared directly. Instead, the hospital runs an RNAseq analysis package on the data, generates gene expression levels, which can then be sent directly to the Treehouse project, and then Treehouse compares that expression profile to other tumors to recommend treatment strategies. One of the rule of thumb on where federated analysis works most effectively is when you have highly identifiable data that yields some form of informative but non-identifiable result. And so the thing that we're working on now is federating co-segregation analysis, in which you analyze together a patient pedigree, variant genotype calls from multiple family members, cancer history of multiple family members, highly identifiable data when it's all put together. What comes out of the analysis is a log likelihood ratio. You're just not going to be able to reproduce all of that data from a log likelihood ratio, not unless you have most of it already to start with. But this log likelihood ratio turns out to be one of the strongest forms of variant interpretation evidence in the ACMG standard. So what does it take to perform federated analysis? There are 4 key components. I won't go into the data because any analysis needs data, not just federated analyses. I'll talk about the other 3 points and the implications for federated analysis. The first is the technology, containers and workflows. And container technology has been a real game changer in this space. So from an IT perspective, a container amounts to a lightweight virtual machine. You can think of it as an image of your software that's translated into a form that can be run reproducibly on all kinds of different computational platforms to get the same result. So if you've ever had the experience of writing some software in R and then discovering that after a new version of R comes out, there's some essential package that you needed that isn't available anymore, or maybe the package is available, but something's changed and you're not getting those same results, that problem goes away with containerization because it bundles together your R script and all the other packages that it depends on. It bundles the installation of R itself, the underlying math libraries, everything that's needed to generate that same result on a different computer. And it literally runs the same on anything from a laptop to a sophisticated cloud machine. And the next piece of it, that are workflows, which basically stitch together multiple containers for a more complex analysis. And these are some of the available technologies. They've all got their pros and cons, but they've all been game changers. This presents the challenge that if someone doesn't trust you with their data, why should they trust you? Why should they trust your software on their system? And it's a very fair question. There's been a lot of movement in this space, a lot of recognition of this problem, and the NHLBI, well, with other funding agencies, is driving a lot of work in container and workflow certification, which is going to help address this in the long term. So while that work is in its early stages, there are some best practices that have emerged. For example, containers should not have any data egress. They write their output to an expected output file when they're done, but don't send any other data out over the internet. And second, the container architecture itself shouldn't require privileged system access. The Docker platform, which is great, well established, does a lot, requires root access. The Singularity platform, which has most of the advantages of Docker, does not. So there are options. Data specification and standards. It should be a no-brainer that when you're writing code to analyze data that you can't actually see, you need clear specifications to tell you exactly what that data looks like. Now, when you're writing, and when you have a good specification from one collaborator, that's a good start, but then as our experience in our consortium, no two collaborators at this time really, manage their data in equivalent data models. So the thing that's going to ultimately address that problem will be data standards. And a lot of that standardization is being driven by the GA4GH. And this is going to be great because as the standards emerge, there will be more tooling around the standards, and that will push more people, well, that will push ultimately adoption of the standards because that will make the most economic sense. So there are legal issues, enough that I've pondered going back to law school to sort this out myself. The short story is that for compliance with the major data privacy laws, HIPAA here in the United States, GDPR in Europe, and similar GDPR-like policies elsewhere in the world, data sharing is, well, federated data sharing is possible because these, by and large, these legal policies address individual level data. If you're doing an analysis that transforms individual data to aggregate data, then that's technically out of, well, that's not in the direct scope of these laws, so it's compliant with the principle. There is still a big regulatory burden on your collaborator to demonstrate compliance with the law, so it's possible but difficult at this time. Just a few months ago, a number of European science agencies wrote a big policy paper on how the GDPR has impacted scientific research and cancer research in particular. The GDPR, incidentally, was never motivated by cancer research. It was motivated to protect against the excesses of big tech in personal data mining. Yet its impact on scientific research has been, well, now loudly recognized. And what I'm hearing from my collaborators on the policy side is that the pendulum has swung, and we're moving into an era where data sharing will probably become easier. So, in conclusion, federated analysis can unlock data from siloed cohorts, and that data can valuably help interpret VUS. A big factor in this is the container technology, which has made the code portable, so you can send a sophisticated analysis package to your collaborator rather than asking your collaborator to do the painful installation process that we've all become familiar with over the years. The biggest challenges at this point aren't technical, but legal and regulatory. At this point, the signs are promising that this will become less challenging in the future. I have many collaborators to thank for this work, particularly the folks in the GA4GH and Enigma Consortium, and my many collaborators in the Bracket Challenge who've been working with me for years in thinking how we could share data on germline variation responsibly. And with that, we've got a few minutes for questions.
Thank you so much for the excellent talk and such a great initiative. So we do have one question from through our virtual platform. Again, starting with a comment, great talk. Can you describe a little more what it looked like to complete the pilot federated analysis with a group in Japan? How much input was needed to run your software on their data, and what kind of training support was required? Was it relatively straightforward? We should add probably what did it take to get regulatory compliance issues figured out?
Absolutely. And let me scroll back to that slide. Yeah, here we are. So, well, for the data specification, in this case, my collaborator at Riken did something which was really clever, which was he generated for us a synthetic data set, describe a synthetic representation of their cohort by effectively shuffling the values in the actual cohort. So that produced a data set where we had accurate representation of the data values, but no data on any single individual. Synthetic data is in general very, very valuable for federated analysis development. What did it take to run it on their side? Well, one of the key things it took was someone from their IT group. As we've been planning the next round of collaborations within our larger consortium, the Enigma Consortium, Momo at Riken has said we could do another federated analysis, but it would have to be a federated analysis because our IT people have some bandwidth, but our grad students and postdocs are all busy. On the regulatory side, well, I'm actually not sure what it took. And so, well, does that address?
Yeah, I think it does answer the question. Actually, we have another question from the audience. So go ahead.
Hi, that was a great talk, and it really talks to many of the big problems in data sharing and trying to address the issues of trying to get information about rare variants that exist in silos around the world. One of the other related challenges as we move into a federated data sharing ecosystem is the notion of overlapping data, patients that are seen at multiple institutions, as we're doing a lot of work to try and, make the data safe and protected and private, et cetera, there's still a need to, I think, share important metadata around the origin of where it came from. Can you speak a little to metadata standards and, you know, this problem of, you know, repeated sampling from individuals who may have been at multiple institutions?
Well, Alex, you've nailed one of the big problems, and that is when you're getting data from multiple sources. How do you guard against redundancies when you can't, you know, when you can't see what individuals the data is coming from? There's been a lot of work on, well, related to COVID, of course, on generating generating cryptographic hash tags from the patient's personal data, and those cryptographic hashes can then be compared and still need to be shared externally, but can be shared externally without directly exposing any of the PII. And so that's one, well, that's a promising direction, not only for finding redundancy across institutions, but even redundancy within single institutions. So one of my testing lab collaborators describes how, well, a number of patients are getting retested these days, particularly patients who'd gotten tested on small test panels back in the past. And if the patient has been retested under a different last name, they can't easily tell that it's the same person. So we've been thinking in terms of a system of cryptographic hashes that uses stable identifiers, first name, date of birth, plus the variant. And so that's what we've got our fingers crossed on.
That's really cool. Thank you.
So unfortunately, we ran out of time. Thank you so much. I'm sure there are many more questions, but we'll have to move on. So thank you very much again for the excellent talk.
Thank you.